The following lesson is the instructional planning laying out of the strategies used in this particular lesson on the structure of DNA. The instructional strategies used in this lesson are audio-visual, whole class, peer-to-peer, kinesthetic independent assessment, as well as reinforcement. We'll go through each of them and how they're involved, but essentially this is a lesson on DNA and nucleotides, the components of them, what makes up nucleic acid, as a lesson that will lead into DNA replication. So as a audio visual for a hook for getting the students interested, there's a brief video called The Twisted Tale of DNA that goes through some of the background of DNA and the bonding that occurs. And then during the whole class discussion, there's some slides that we'd work through with the PowerPoint that go through a few subtopics. First is nucleic acids, their structure and their function, dealing with what nucleic acids are, the different kinds there are between DNA and RNA, and the subpieces of those nucleotides, the nitrogenous bases and the pentose sugar and the phosphate group. The next component of it is the bonding with the phosphodiester bonds and the van der Waals forces between the nucleotides. So the A to T, C to G ratios of adenine, thymine, guanine, and cytosine, and the number of hydrogen bonds that make them unique and only bonding to each other. And then the final component of the whole class discussion portion is on the discovery of the shape of DNA and its hints to replication, in that it's a complementary strand that's running anti-parallel, two different strands, and then the implications of that, suggesting for replication and between Watson and Crick and Rosalind Franklin's contributions. Through this portion, the students are asking questions and also answering questions that the instructor is asking. But on top of that, they are expected to be taking notes during this that they would then use, they would then pair up peer-to-peer to work through this graphic organizer, going through the two different types of nucleic acids of DNA, RNA, and identifying the structure and function of nucleic acids. Then the kinesthetic portion is the model building. So what the students would do, this is it's a pretty low budget model, which is nice because they can all be made with simple products that you can get from any store. Masking tape, Sharpies, colored pipe cleaner, where they will build essentially a model of DNA. And what I have them do is, you can see here, I have the carbons, the 5 primes, all labeled. I have them label, I have each student label one of each, but they're building this and they identify which of the purines, which of the pyrimidines. They make an index card as a key and color code them. So the A's, T's, C's, and G's are unique. I encourage the students to choose colors that make sense. My wife is a Florida grad and they're University of Florida and their colors are orange and blue. Or I'm a Florida State grad. The colors are kind of garnet and gold. So A's only go with T's, C's only go with G's. So I encourage the students to choose colors that make sense and match for them. So this is really the building component, and it's really where they fine tune, at least identifying the sugar phosphate backbone between the bonding at the atomic level. Now the independent assessment portion of this is that with them working in pairs building this model, one student is able to draw the sugar phosphate backbone on their model on one strand, where the other student's able to build and draw and label the complementary strand. So between the two, they each have their own independent strand that is a complement of the other. So even though the two of them are building a model together, they both have independent, they're able to be assessed independently. And what is this reinforcing, or what is this getting back into? How do we reinforce it? The following day, they will use their models. They will unzip their models and they will use it to then replicate that DNA using the same techniques that we used before. But in this, we'll be able to address the leading strand that's continuously developing as it unzips. And then the lagging strand with Okazaki fragments, discussing the features that come after that. So essentially what we have are several different instructional strategies that are all used within the one lesson, designed really to tap into several different types of learning styles. I hope this helps.